We can get going, I guess. So yeah, welcome, good morning, good afternoon, or good evening, wherever you're sort of tuning in from. Thanks for joining. So for anyone that is attending this call for the first time, we've been holding these industry-wide calls every month for really any company that wishes to participate in the cultivated meat value chain. And the purpose of these calls is really to sort of introduce and welcome new solutions providers into the industry with the end goal of hopefully establishing new collaborations that can help accelerate R&D and commercialization in the field. So if your group is interested in presenting on a future call, or maybe you're working with a company that could benefit from being on these calls, please feel free to put them in touch with me directly. And as always, the sort of sign up link to this seminar can be shared with other colleagues in your company if you want to sort of have them join and tune into these meetings in the future. So just some housekeeping items from our side. I hope everyone can just keep an eye out for the Bezos Earth Fund's AI Challenge. One of those focus areas is going to be on sustainable protein. And it would really be great to see some cultivated meat work get funded through that area. So I think we sort of are anticipating, based on their website, that an announcement will sort of be soon for the details of this, but I'll drop a link in the chat. They also have a sort of sign up box at the bottom of this page that you can receive updates, but we'll also be sharing the information when it becomes available to all of you. So if you're working sort of on AI related tasks within your company, this might be good to start thinking about ahead of time. Also, I just wanted to give a heads up that our July seminar is going to be hosted by our GFI Asia Pacific team. And so that will be taking place at a different time. So we're going to sort of trial this to accommodate our audiences that live in different time zones, and we'll likely have more instances like this in the future. But just wanted to sort of give you a heads up on that. Yeah, I think some calendar updates will be coming your way. And with that, that's all from me. So today, pleased to welcome Tatiana Cucu, who will be sharing information about their services in aroma and end product analysis. So Tatiana, I will turn it over to you.
Okay, hello everybody. First of all, I would like to thank Renee and Elliot for giving us the possibility to present today. And what I would like to talk about today is actually about aroma profiling and how this can support the industry, which is doing quite some innovation nowadays. But before I will get into that, I would like to introduce you to RIC group, RIC Resolve. So who is RIC? RIC is a privately owned European Center of Excellence in the field of analytical solutions. And these solutions are mainly based on chromatography and mass spectrometry. And we are supporting customers from different industries, ranging from food, feed, pharma, petrochemistry, and other industries with scientific know-how and technical expertise for already more than 35 years. Our main offices are located in Belgium, in Kortrijk, where actually also our laboratories are located. And the services that we are providing to our customers are ranging from method development to characterization, consultancy, training, and we are even helping our customers when they get some SOS cases. When it comes to the analytics that we have in-house, these are actually divided in many parts. We are working a lot on sample preparation. So basically everything from extraction to cleanup, pre-concentration. And we have the experience of working with sample amounts which are very big. But in case of cell culture industry, of course, we have to also learn to work with small amounts of material. And there we also have experience with that. We work also on separation, and the separation that we are doing is mainly gas chromatography or liquid chromatography based. And this in combination with different detectors, which are ranging from mass detectors to FID detectors, UV detectors, and so on. And these all being state-of-the-art equipment. And of course, the data and the reports that we are providing to our customers are depending a little bit on the question that we have. but this can be, for instance, quantification reports, it can be identification reports, and even where needed, we can support our customers with quite some statistical analysis to help find some trends in the data or highlight differences between different samples, for instance. Now, if you take a look at our portfolio, you will see that we are actually active in many domains. We are doing proteomics, lipidomics, we do process monitoring, lipid residual testing, and so on. But the presentation today will be dedicated to aroma analysis. And why aroma analysis? Well, because especially in food and beverages industry, the aroma plays a crucial role. We know that the overall acceptance of the products by the consumers is related to the aroma. Consumers tend to perceive the quality of the food by its aroma. Something that smells good is typically perceived as of a very high quality. And that is why aroma analysis is actually of a paramount importance for the food and beverage industry, and this at different levels. At the raw material selection stage, at the product development stage, also when we want to formulate products, we need to make sure that these are products which are having consistency. So if we make different batches of a certain product, this has to be consistent today and tomorrow and within 1 year. We are also applying aroma analysis when we look into stability of the food products or beverage products. We look at the stability during processing, but also during the storage. Is the aroma still stable, or is there any off-flavor compounds formed? So this is also a very important parameter. Now, when it comes to the switch from the conventional foods to alternative and sustainable foods, of course, aroma analysis has proven to be here also very important. Now, despite the fact that when we talk about aroma compounds, we know that these are very small molecules, nonetheless, they have a big impact on the product, but often also on the commercial value or the market value that we can give to a certain product. And here, just a very small example for you. So like we know, there are rice varieties, and rice in general contains about 92% starch and 7% protein. But the huge difference in the market value, it's actually related to the fact to the remaining 1% of volatile organic compounds or the aroma compounds. So aroma is very important for the food and beverages industry. And in order to be able to perform good qualitative aroma profiling, we depend on several factors. Actually, we depend on 4 factors. First of all, sample preparation techniques, how we will extract the aroma from the food product. After that, separation method. Which type of separation are we going to apply? Are we going to do a classical one-dimensional separation or a more complex multidimensional separation to get even more information about the complexity of the aroma that we are analyzing? After that, we also need to combine that with proper detection techniques. And of course, it's interesting to have the mass detectors which can give us information on the identity of the compounds. It's also sometimes important to be able to do a parallel olfactory detection, because let's not forget, the human nose is a very interesting and very sensitive detector as well. Now, finally, as
the red ones, yeah.
Yeah, sorry. Okay, and then when we perform aroma profiling, typically this means that we are going to have many compounds analyzed at once. And that is the reason why after all this work, actually, we need to have proper data processing tools which can allow us to get the right information out from the analysis that we perform. And this can be, for instance, statistical analysis, or for example, combine or search in aroma databases to find better links between different compounds. So let's start step by step when it comes to aroma profiling. So as I said, sample preparation is very important. And when it comes to aroma profiling, typically we talk about untargeted aroma profiling, which means that we are not looking at 1 or 2 compounds which are present. What I often say, we are not searching for the needle in the haystack. but we actually want to know what is the composition of the haystack. So this means that when we want to do aroma profiling, we need to have extraction methods, aroma sampling methods, which are non-specific and allowing us to detect the broadest range of compounds at once. This means that we want to be able to identify or analyze the volatile compounds up to non-volatile. So in one analysis, one would like to see the presence of methantiol if present, but also the presence of vanillin, which is a semi-volatile. We also, of course, need to have a very high sensitivity extraction method. So not all methods are able to extract, extraction methods are able to extract the aroma compounds even good. And we also prefer to have methods which are involving minimum sample preparation. Why? Because we know that during sample preparation, some compounds, some aroma compounds can be lost, or even some artifact compounds can be formed, which can give us wrong information about the sample. And that is the reason why we're actually opting to use green, automated, fast, and simple methods. And when we talk about these type of methods, actually we are coming into the range of headspace techniques. What does it actually mean? It means that when we analyze aroma, we will only analyze the volatile part of our product. And to do that, we have a whole range of headspace extraction techniques. And these methods can range from low sensitivity methods up to very high sensitivity methods. And depending on the method that we apply, we can have a more discriminative or non-discriminative character. What does it mean? At headspace analysis, for instance, if we talk about static headspace analysis, we typically what we do, we place a sample in a vial, we incubate the sample at a certain temperature, and we wait until there is an equilibrium between the sample and the headspace in the vial. Once this equilibrium is reached, what we are going to do, we will inject a small portion of the headspace, and we will analyze which compounds, which volatile aroma compounds are present in the headspace of the sample. We can do that with an alternative method, for instance, with SPME, solid phase microextraction techniques. In that case, we also have a sample which is introduced in the vial and is equilibrated. But instead of injecting the headspace, we are introducing a fiber, a small polymer fiber of different chemistries. This can be of different chemistries. And what we are going to do, actually, the volatile aroma compounds will get adsorbed on this fiber. And after that, what we are going to do, we will analyze the fiber by introducing into the GCMS analyzer. The difference between the static headspace and the SPME technique is, in fact, that depending on the chemistry of the fiber, we might have a better selectivity or a better extraction for one group of compounds and less extraction for a different group of compounds. Then we also look more into actually, our preferred method of analysis is dynamic headspace analysis, DHS techniques, including multi-volatile extraction techniques. Why do we prefer the dynamic headspace techniques? Is because these are the most exhaustive extraction techniques that we can apply. And in fact, we have again a sample in a vial which is incubated at a certain temperature. And instead of waiting until the equilibrium is reached between the sample and the headspace, we are going to flush the vial with an inert gas, and we are going to sweep or to actually extract exhaustively the aroma compounds from the sample, and we will trap this on a special trap, which is later on analyzed via GCMS. And just to give you a small example on how big the difference is between the different extraction techniques, here we have an example of an typical food aroma mixture, which contains more than 100 different compounds of different chemical families. And if we analyze the same aroma mix via DHS dynamic headspace extraction technique or solid phase microextraction, you already see that via SPME, we are not able to analyze the very volatile compound. So a lot of information will be lost. Why? Because we are confronted with the selectivity of the fiber that we are using to do this analysis. On the other hand, we will also lose some information on the semi-volatile compounds. while the dynamic headspace analysis is actually providing us with the most complete profile possible from the very volatile up to non-volatile or semi-volatile compounds. But this is, of course, a mixture of aroma compounds. Let's look more into details into a food product. And here we did a comparison between the dynamic headspace analysis and SPME analysis. What it was interesting to see is that by applying dynamic headspace analysis, we could detect 2 times more aroma compounds compared to SPME. For instance, we could not detect the 2-nonenal, which is known to give an unpleasant tallow aroma. We were not able to detect via SPME extraction, but we were able to detect that via dynamic headspace, and also some very interesting nutty aroma compounds which were detected via dynamic headspace, but they were not detected via DHS. So actually, selection of the headspace technique for the aroma analysis is very, very important. But actually, with the instrumentation and with the modules that we have on our GCMS systems, we are actually able to go even a little bit further than that. We are even able to simulate cooking, for instance, by incubating our samples for very exact temperature or at very strict temperatures and for very so for the exact times. For instance, this was a test in which we placed 0.2 milliliter of oil in a vial. We added 3 grams of sample, and after that, what we did, we transferred this sample to an agitator which was hot at 100 degrees. We simulated the cooking for exactly 1 minute and 50 seconds, after which we could compare the aroma profile of different samples by analysis via headspace. So you see here, for instance, it was a cooked, let's say a simulated cooked conventional burger and plant-based burger, which was cooked in our agitator, and after that we analyzed the aroma. And like this, we can already see that there are several compounds which are similar in the conventional burger and the plant-based burger, but there are still a lot of differences when we look into aroma, differences between the different types of products. How about cooking during sampling during cooking? Well, this is also actually possible. We are even able now to sample the air or the aroma which is released during cooking by concentrating this aroma on special traps and then analyzing this aroma on GCMS. And we did this actually to analyze some plant-based burgers, and it was very interesting to see the profile of the burger itself, so after baking, and then analyzing the aroma which is released during the cooking. You see the profiles are very complex. We have many compounds which we are able to detect in there. And in fact, if we are going to look into more details into compounds which are responsible for the pleasant roasted, grilled odors, then you will see that we are able to detect these compounds not only on the burger itself, but we are also able to detect these compounds in the aroma which is released during cooking. For instance, we saw the methylpyrazine, the 3-methylpyrazine, and some other ethyl-methylpyrazine isomers. So this can also all be automated or all be simulated and then analyzed via GCMS. A second thing when it comes to aroma analysis is separation. And in fact, we always want more. We want very complex sample analysis in one single run. Unfortunately, what we have in that case, we have very complex samples. We have a huge number of constituents in one single run. Additionally to that, we have huge differences in the concentration ranges. Sometimes we have aroma compounds which are present at ppb to ppt levels, while others present at ppm to % levels. And also we have compounds which are belonging to a whole range of different chemical classes. So this is becoming very complex. Of course, one way to avoid that we have too many coelutions or too many compounds eluting together would be by simply slowing down a oven gradient. And that will, as in the example here, already help us to resolve more compounds, because if compounds are coeluting, we are not able to identify them correctly. So we really need to have a good resolution. Now, you see, in this case, by changing from 5 degrees per minute to 2 degrees per minute, in this region, we could get better resolution. But we still have regions in our chromatograms where we know they are very crowded, and we know that we still have a lot of coelutions. So what to do about that? Well, in order to overcome this problem, we are actually also switching regularly to multidimensional chromatography. So what does multidimensional chromatography means? It means that we are combining different chromatographic separation mechanisms in order to improve the separation, in order to resolve compounds which are coeluting in one single dimension. And to do that, we have the possibility, we can do that via different ways. In-house, we are able to apply hard cutting techniques. So basically, when we do GC for highest resolution possible, or we can apply comprehensive techniques. This means GC times GC, where we are able to get the ultimate resolution. And I think these names maybe don't mean much to you now, but I would like to explain that to you how it is done. So basically, when we talk about hard cutting technique, it means that when we have an analysis in one dimension, so we have a chromatogram, we are going to choose a certain region in the chromatogram, and we are going to analyze it on a second column, which is of a different polarity. This means that we will be able, like that, to resolve more compounds, so to remove or separate more compounds in our samples. Actually, these techniques has proven to be very useful, especially in the off-flavor monitoring. Why? Because we know that off-flavor compounds are often present at very, very, very low levels. Often the human nose is able to detect them, but we are not able to identify them easily with GCMS techniques. So by applying this hard cutting technique, what we do, we are able to separate the off-flavor from the matrix compound, and like this, we are able to, as I call it here, find the needle in the haystack. But actually, we can do a bit more than that. We are actually able to do comprehensive GC times GC analysis, in which instead of just analyzing one zone via second dimension column, we are analyzing every single piece of our one-dimensional column on a second column, shorter column of a different polarity. Like this, allowing us to gain much more resolution, and often in one apparent peak in one dimension, we will not be surprised to find 2, 3, or even 4 coeluting aroma compounds. And this is an example here of a very complex product which we analyzed, and which we, in one dimension, one analyzing by one dimension, we were only able to detect pyridine because this was more abundant. But when we applied the comprehensive technique, we were able to detect also dimethyl disulfide and 2-butanol 2-methyl, which are very important aroma compounds, especially the dimethyl disulfide, which was not able to be detected in the conventional one-dimensional analysis. Just to give you an idea, as a rule of the thumb, by switching from one-dimensional chromatography to comprehensive chromatography, typically we gain about 10 times more information. And this is an analysis of a product which we did, first of all, via one-dimensional analysis, classical one-dimensional analysis. And what we were able to do there, we actually were able to identify between 353 to 380 different aroma compounds. When we analyzed the same compound via GC times GC or comprehensive GC, we were able to detect more than 3,000 compounds. So you can see that by adapting our separation technique, we can also get more information about aroma compounds. Now, of course, detection is also very, yeah.
I just want to give a note on time, just to make sure.
Yeah.
We got about 5 minutes or so.
Yeah. So for the detection, of course, this is also very important. We have different detection techniques in-house, this ranging from single quad analysis. Of course, this one is very robust and is a lot. but we know that this is slow and it does not allow to identify many compounds, especially when they are coeluting. We have FID detection, which is allowing to do quantification, but no identification. And our preferred technique to do aroma analysis is TOF detection because of its known sensitivity and also because we are able to do fast acquisition, which means that we are able to get more untargeted information, so more compounds are identified at the same time. And here an example of an analysis which was performed with conventional food products, in which we did an analysis with a single quad analyzer and a TOF analyzer. And you see that typically by switching from one type of detection to another, we are able to get at least 2 times more information. You see here in an yogurt or a wine sample, and even also higher sensitivity, because if we look into signal to noise ratio of the single quad analyzer compared to the TOF analyzer, we have here much higher responses for compounds which are known to be very important aroma compounds or aroma characteristics, such as dimethyl disulfide or a hyptanone or a hyptanal, which have very characteristic odor. Even in very, very complex compound samples where we have compounds which can interfere with or matrix compounds which can interfere with the aroma. For instance, we are even able to detect important aroma characteristics, such as, for instance, here metional, which is known to have a baked potato aroma, which was present at very, very low levels under the acetic acid peak, and still we were able to detect via TOF analysis. We combine our analysis often with retention index confirmation. Why? Because we know that when it comes to aroma, often the spectra that we get, so the spectral information that we get is not very selective. So we always compare the retention index with calibration, and we compare the retention indices. So for an apparent similar spectra, we are able to quickly find what is the difference between a 2-heptanone and 2-nonanone, thanks to this information. Finally, as I said, we have mass analyzers which are helping us to identify compounds in a sample, but often the compounds are present in the samples at levels which are not detectable by the state-of-the-art instrumentation. So it's very interesting to combine that also with synchronized olfactory detection, and that we do that with a special tool which allows us to do simultaneously a GCMS analysis for identification and a sniffing by a panelist, so that important aroma regions can be detected in our sample. Finally, we have also a very big aroma database, so a database which contains information on odor characteristics or typical odor compounds from food products. So we can easily search in that database, and if we sniff something in a sample, we can try to find what is the compound which can be responsible for a certain aroma characteristic. And finally, we are even doing quite extensive statistical analysis, because data, as you have seen the chromatograms, when it comes to aroma analysis, we have very complex data sets, we have complex chromatograms, so it's difficult to find trends in our samples. So we have special software packages which are allowing us to, for instance, on one hand, do principal component analysis, where we can see the difference between samples. We can look at compound level to see which compounds are similar, which compounds are different, and also see the trends between the different samples. For instance, here we saw that hexanoic acid was more abundant in a conventional sample, burger sample, while less abundant in a plant-based sample, and the vice versa was the case with the furanol. And these are compounds which are very important in food products. Okay, I hope I didn't go too fast, but nonetheless, I would like to take 1 more minute to thank some people which have, of course, contributed to this work. Our GC geek, which is Frank, our team of scientists. We have 2 inspiring supervisors, Pat and Kun Sandra, father and son. We also want to thank our clients and partners, which are making sure that we are going the extra mile also in aroma analysis, and also Good Food Institute for the organization of this session. If you have any question, please feel free.
Thank you so much for that presentation. This is far outside of my wheelhouse, so I think we have time for maybe one or two quick questions if anyone has any. Awesome. Well, we're at the top of the hour, so thank you to both presenters today. This is a really interesting group of talks. I hope some folks will reach out to you in response to this, and we will see you all here next month.